Good morning, everyone, or good afternoon, wherever you're located. Thank you for joining us. My name is Marissa Waterman. I'm the Marketing Director here at the Academy. Here with us today, we have Dr. Sandeep Sathamurthy, a global practice and technology leader of innovation and applied research at one of our patron companies, Black Veatch. Today, Sandeep will be discussing innovative platform MABR Research Collaborative, which won the grand prize in our research category of the 2021 AAEES Excellence in Environmental Engineering and Science Awards. Before we get started, I just want to mention that you'll be able to message your questions at the end of the presentation. Once the presentation's over, you'll be able to type your question into the attendee chat panel on the left-hand side of your screen. Okay, let's get started. Welcome, Sandeep. Thank you for joining us today. We're ready when you are.
Thank you so much. Thanks everybody for being on. I will, if you don't mind, turn my camera off. That way the slides are a little bit larger. So as Marissa mentioned, we are going to be talking about our collaborative research with the City of Hayward and Suez. Super excited to share our results with you, but I wanted to start by contextualizing it a little bit. The why did we think this is an important piece of work, for us as well as for the community more broadly. So I want to talk about sort of the roadmap, or what we see are the drivers for some of the changes that we're seeing in the development of infrastructure today. I think all of us are experiencing these mega trends: climate change, urbanization, scarcity of resources, and not the least is the ability to get data much more easily and much more cost effectively. And there are others, but these four seem to be driving a pretty significant degree of change within the infrastructure community, and especially as we think about the transition from traditional wastewater treatment to integrated resource recovery. Again, I think all the utilities are experiencing these in very different ways. And the box that I'd like to focus on a little bit is the second from the right, which is the process intensification box. We see this happening a lot in our systems, where more and more the value of intensifying our processes is fairly tremendous. What is process intensification? Just in a word, it's effectively doing more stuff, more things, more capacity, more treatment with less infrastructure or equipment. More specifically, you can see it as taking what assets you may have and through the use of technology, innovative technology, using the same assets to accomplish either better or higher treatment goals or treat a higher capacity. So I like this graphic a lot. It speaks to me a little bit. Just it's using less energy, using less chemicals, making things smaller, and getting out of it more value, more capacity, and more stuff I can make things with. Whether that's energy, fertilizers, whatever those things are, I get more of them. So as we drill down a little bit more into the world of nutrient removal, many of our utilities across the globe and in the US are faced with aging assets and still need to meet increasingly stringent nutrient requirements. So there's this array of intensification technologies that are being applied. Certainly one can build more tanks in certain configurations, but there are more and more technological devices to use. The one we'll be focused on today is a biofilm system. So I've classified it here, and it's the membrane aerated biofilm reactor. It's in the second box, and it's the second box in the second box. So it's a counter-diffusional biofilm system, and we'll talk a little bit about what that means. So that's the focus of our presentation. So what is a membrane aerated biofilm reactor, MABR? Essentially, you take a membrane, which we're quite familiar with for many years now. You combine that with not filtration, but rather gas, and I'm going to focus it on air delivery within the membrane itself that's being pushed out into a biofilm on the outside. I'll show you that in a moment. You modularize this into some cassettes, and what you get, and I love this phrase, it's one of my colleagues has used a lot. You get a breathing biofilm. Essentially, you've got bacteria growing on the surface of the membrane, and I can control not only what gas I send, I'm using air, but also how much of it I send and go directly to the biofilm itself, directly to the bugs, rather than from a air bubble, into the bacteria through various mass transfer limitations that we've experienced over the history of wastewater treatment. So the core result of this is I get engineered biofilms at a location where I want them, doing the work I want them to do. And they're breathing without any oxygen in the liquid environment itself because it's coming from the MABR. So here's a little bit of a detailed view of what's going on within the lumen and the biofilm itself. So looking at this from left to right, the white is inside the lumen itself, and the gray is the membrane wall. Now, to be sure, these are not filtration membranes, so they don't allow water to. It's not the intent for them to enable water to filter through. Rather, they're specifically designed materials to enable a high diffusion of oxygen or the gas of choice. Again, I'll focus on oxygen to diffuse through the membrane into the biofilm itself. So what I'm showing here is really focused on nitrogen management coupled with COD removal. So you have in the bulk liquid on the far right-hand side of the graphic, you have ammonia and COD or BOD and so on. They're diffusing into this biofilm that's growing. And on the left side, you have oxygen that's going in. They both meet somewhere. There's a diffusion gradient for both of them. And this counter diffusional arrangement where the oxygen and ammonia are coming from separate sides results in some unique biofilm behaviors. And this is very different from the historical biofilms that we've applied within wastewater treatment, whether trickling filters, RBCs, biologically aerated filters, and so on. So here you have the substrate and the electron acceptor coming from opposite sides into the film. In this particular graphic on the right, you'd have a very high ammonia concentration in the dark brown zone, and on the left you'd have a very high oxygen concentration in the light brown zone. And somewhere in the middle, you'd have a lowish concentration of both. Not exactly at this hard line I've shown, but somewhere in the middle there. Consequently, you have an aerobic portion of the biofilm on the inside, lots of oxygen, some ammonia, and you've got a low oxygen, so anoxic or anaerobic, depending on the redox condition, environment on the outside of the film. So you can accomplish nitrification, so conversion of ammonia to nitrate and nitrite on the inside of the film. And you can potentially accomplish denitrification, that's conversion of the reduced nitrogen species, nitrite and nitrate, into nitrogen gas using the COD or BOD in the bulk liquid itself. So in a single system, we can have both bubbleless aeration. So again, no aeration through a membrane diffuser, which is what we've used for a very long time in our domain, and a biofilm technology. So it's quite an interesting combination of these two functionalities that gives you, very efficient aeration coupled with specific bugs of interest within the membrane biofilm itself. What is the end use value and proposition? Of course, it's about reducing the costs, so the capex, as well as the operational cost of accomplishing whatever the treatment goals are. So here's just an example. Here's a conventional MLE process with an anoxic zone shown with this mixer on the left of the reactor, and then a long aerobic zone. If you converted this into an MABR suspended growth hybrid, you would essentially put the membrane aerated biofilm reactor into the anoxic zone. And here it achieves both nitrification and denitrification in that same anoxic reactor. So your suspended growth system is doing a little bit less work. There are at the moment, three very prominent technology providers. They've all been around since sort of the early 2000s, so coming on a while now, but not that many references in the US anyway. It's growing. There are an increasing number, but it's not ubiquitous. Now, despite having this longish history of technologies in place, there remains some very key knowledge gaps. So going from left to right, we really are just exploring and starting to understand how does the microbial community within the biofilm itself change as a function of a variety of things, including the use case itself, the oxygen concentrations, the pressures, the bulk conditions, and so on. What is the ratio of ammonia oxidizing to nitrite oxidizing bacteria? And even more broadly, what is the function that's engendered in the biofilm? Then going to the right of this graphic, there are also some pretty practical questions that still need some exploring. And some of them still do at the time of our research. How do you design these? What's the best use case for them? Where should we put them? Where should we locate it? What can I assume for a nitrification rate, which will impact size not just of the membrane aerated biofilm reactor, but everything downstream as well as blowers and so on. So this kind of exploration of the knowledge gaps is what really led us to want to work on this research project a few years ago now. So this is the meat now of what I'm going to be presenting. It's our collaboration over a couple of years with the City of Hayward and Suez. What I'm showing here is a culmination of a lot of folks' work, and I'm really appreciative for the whole team's effort, engagement, involvement, and just enthusiasm to do this, to do the research. A little show and tell. Here's the pilot at the Hayward plant. You're seeing the Hayward plant in the background. It's about a 10 mgd facility, fairly conventional, has a trickling filter and solids contact tank configuration. This is actually a Google Earth image, and I guess they flew their satellite when we had the pilot there. So now we've really made it in the world. We've shown up on Google Earth. A little bit of a view on the right-hand side of the inside of the pilot. It's quite small. You really have to squint, but there's a little zone to the left of that blue vertical line where the membranes are. There's a small anaerobic anoxic zone, then the membranes themselves, and two aerobic zones, kind of to simulate a treatment plant environment within a selector, anoxic zone, and then an aerobic zone downstream. So we broke our research up into three key questions that we were trying to understand. And our team, Black Veatch, the City of Hayward, and Suez very much focused on the applied part of applied research, trying to understand, okay, does this fit not just for the city of Hayward as a potential option, but more broadly in the San Francisco Bay Area, where a number of utilities are trying to figure out what is the path forward in terms of nutrient removal. So the three core questions that we were trying to answer are outlined here. And essentially, it's about how big, how much membrane area do I need, rates, and can I use this in any given configuration? So usability, feasibility. Well, what I'd like to do is to go through what we explored as answers and how we addressed each of these questions in turn, and share the results with you. So how our first question is all about, okay, how much SRT? We use this as a parameter to classify our treatment systems, and we have some very good understanding of how long an SRT do we need to achieve nitrification. We did some modeling work. In fact, there's a paper. The first author is one of my good colleagues, Kelly Gordon. We presented it at WEFTEC a few years ago, where we explored in a modeling framework what is the potential value proposition. And so here you're seeing some results from that. So if you look at the plot, essentially what this is saying, if I draw sort of a horizontal line at around 0.5 milligram per liter ammonia, if that's my target, if I'm using a conventional MLE process, the green diamonds, I might design for an SRT of maybe 6 days to be safe, or 5.5, something along those lines for this particular scenario. In contrast, if I use a MABR suspended growth hybrid, the circles now, and I want to achieve the same target, this is suggesting that I might design for 4.5 days, or if I want to be a little more aggressive, maybe push to 4 days. Hard to say. One way or the other, there is about a day to a day and a half reduction in the SRT required. This is what the modeling data suggest. And the hypothesis is that it's because of a seeding effect from the biofilm into the mixed liquor, or because the biofilm, the MABR biofilm, is doing all this work, there's less work that's needed in the suspended growth system, so you can make that a lot smaller. And this is primarily what we explored. So we ran the pilot for a long time, and at 3 different SRTs, treating primary effluent from the plant. And at each SRT condition, we ran it for at least 5 of those SRTs before we did an intensive 24-hour composite sampling across the pilot itself. So you're seeing here data from these 3 SRTs, and they're suspended SRTs, meaning only talking about what the mixed liquor concentration in suspension, but not accounting for the biofilm bacterial mass in the SRT calculation. 0.3 days, 1.5 days, and 3 days. I just want to go back for a moment to the previous slide, and you'll see here each of these SRTs is much, much lower than what one would conventionally use in a nutrient removal system to achieve these very low ammonia. So we were, targeting an ammonia somewhere in the 1 milligram and per liter range. We said let's go all the way to around 3 days suspended SRT. So I'm going to show a couple of these plots again from top to bottom. It's increasing SRT. This particular one is for COD removal across the system. So coming into the pilot, let's say around 300, and leaving the MABR at the shortest SRT around 100, and going across into the effluent around that same 100 or so. We're definitely getting some removal across the MABR itself, but none in the suspended growth part of it. As I increase the SRT, that particular trend that we're not getting any more COD removal across the suspended growth piece of the pilot remains fairly consistent. But as we'd expect, as you increase the SRT, the effluent COD goes down to around 50 milligram per liter, and that doesn't seem to change very much. Not a great revelatory result, but interesting to see that even at a very low SRT, we can get quite a lot of BOD COD removal. Here we're looking at nitrogen species now. So the blue are ammonia, red are nitrite, and the green are nitrate concentrations. Again, left to right is going from the feed of the pilot to the effluent of the pilot, and top to bottom in the panels is increasing SRT. Here, I actually want to start at the bottom. So at a 3-day SRT, where the ammonia coming in, and it's all largely ammonia, let's just say in round numbers is in the 40-ish milligram per liter range. Leaving the MABR zone, the ammonia is down to around 15, with very little nitrite and very little nitrate at a 3-day SRT. So we've lost nitrogen, gone to nitrogen gas, and then we see that in the suspended growth portion, the fine bubble aerobic portion, we're converting more of the ammonia into nitrate. As I reduce the SRT, so as I walk up in the panels, I just want to draw your attention to what we thought was very interesting. At an SRT of less than half a day, and this is less than the SRT that a typical even BOD removal system is operated at, we are getting total nitrogen removal across the MABR zone to the tune of about 40%. This was quite interesting for us. We were surprised, and when we saw the COD result, it sort of adds up. So what this is saying is, with almost no biomass in the suspended growth portion of an MABR suspended growth process, we can get quite good ammonia removal and total nitrogen, meaning we're not making inorganic nitrogen from it. It's all going to nitrogen gas. So coming back to what I showed you earlier, we are indeed getting total nitrogen removal in the MABR anoxic zone. That was really, we were pretty tickled with this result. And then even at a 3-day SRT, so much lower than what one would normally run a suspended growth system to achieve nitrification, we can get excellent nitrification. And with a little supplemental carbon, you can probably drop that nitrate down to something quite low. So a very good result over there. So it does indeed accomplish nitrogen removal. We explored a little bit the biofilm characteristics as well as the mixed liquor characteristics in terms of which bacteria are present. So we used qPCR to assess the total bacterial concentrations or mass or gene copies, and compared that with the known nitrifying bacteria concentration. Shown on the far left is the seed biomass. We had seeded this pilot with mixed liquor, with RAS actually, from a plant that runs very, very conventionally, around an 8 to 10 day aerobic SRT, so nitrifies very well. And then here the data are a little jumbled. They're shown chronologically because of the way we operated the pilot. So second from the left are the data from the 0.3 day SRT. Essentially, there's very little mixed liquor. It's mostly on the biofilm itself. Then is the 3-day, and then is the 1.5 day. So this is quite interesting. We do see that there's at the 1.5 day there seem to be quite the concentration, effectively, or percentage, I'll say, of nitrifiers in the bacterial community is comparable to that of the seed, but we see it's a bit lower in the 3-day. There are reasonably large error bounds around these green bars, and all this is saying, I think, is that the history of the biofilm itself plays a role in in modulating the presence or absence of certain bacteria. What is maybe a big takeaway for us is that indeed the MABR biofilm is quite rich in nitrifying bacteria and comparable to that of a 8 to 10 day suspended growth system. Not 10%, 20%, but in that 1.5 to 3% range. So our second question now, moving on, all about the rates. Of course, this is a membrane system, so the way the metric we use for rates and sizing is the membrane specific rate. So how many grams of nitrogen can I remove or oxidize per day per square meter of MABR membrane surface area? Here we had hypothesized that it should be somewhat node, whereas I have more and more ammonia in my bulk. I should be able to drive to higher concentrations, and this will be a little bit modulated by the DO and a few other conditions. So this is what we had expected. What we saw surprised us a little bit. So you're seeing here just dots of the rates. So the rates are typically in the range of 1 to about 5, we'll say, grams of nitrogen removed per square meter of membrane surface area per day. Right. The green dots are the longest SRT. The blue dots are the shortest SRT, and the orange are the ones in the middle. There certainly does seem to be some grouping based on the SRT itself. Again, some of that might be the history of the film. We'll talk about that in a moment. But what was interesting for us when we look at this plot, especially at the higher ammonia concentrations, where we had expected rates to plateau out around the 3, 4 or some number, it's shown by our dashed line there, and that's our conceptual model of the world. In fact, what we saw is that rates were decreasing. So at very high ammonia concentration, we saw lower rates than at the mid-range, say 10 to 20, and these are concentrations in the MABR zone. We were very curious about this. Why is this happening? What's the mechanism to explain this? So we have a couple of different hypotheses that we explored. And one of the things that we used to inform that, we also looked at the oxygen transfer rates. We were able to measure those. So we know how many grams per square meter of membrane surface area per day that we're providing. We know theoretically what it should be. These are the blue lines. So the dark blue line is for nitrification, so going all the way from ammonia to nitrate, and the lighter blue line is for nitritation, going from ammonia to nitrite. So we looked at this as a function of the ammonia removal rate itself, and what these data show is that at lower ammonia removal rates, so the orange dots here, the 1.5 day SRT, something else seems to be using up oxygen, resulting in a much higher oxygen transfer rate than we would expect. So what else is going on? We said, well, it must be for BOD removal itself. So the first hypothesis that we considered is, look, if you're running at a shorter SRT, you have less nitrates returned and less of the BOD or COD used for denitrification. And so more of it is oxidized aerobically in the biofilm itself. So that's why, as you go to the shorter SRT, so it's an SRT function, you're using more of the oxygen to do BOD removal in the biofilm rather than nitrogen removal. So we did indeed explore that, and yeah, there seems to be something useful there that we think has some value for us to explore, because we see there is indeed a bit of a relation there. The other hypothesis that we explored is maybe it's just time. Like any other pilots, we were not doing lab scale assessments, so we're not controlling the feed. It's whatever came into the Hayward plant and whatever came out as PE, that's what we were treating. And you can see that we jumped around. So we did the very short SRT first, then the longest, and then the mid-range. So maybe it's just time and the effect of time on the film itself. And that one, it's much harder for us to say yes it is or no it's not. We think it's some combination of both. However, our data do indeed seem to suggest that there is a role that SRT plays, the suspended SRT plays, in mediating the community within the MABR biofilm that will influence how oxygen is used and what the functionality, so what the ammonia removal rates are in the biofilm itself. The third question that we were keen to understand is where can I use the MABR? And really, as we thought about this, we were thinking about the impact of carbon to nitrogen loading rates. So if we think about a treatment facility, in the influent, you've got a lot of BOD, COD, as well as a lot of ammonia or organic nitrogen. If I go to a primary effluent, I've removed some of my BOD, COD, the particulate fraction or some of it, but I've not removed any of the ammonia. So I've changed the carbon, and here we use carbon to mean organic carbon, to nitrogen ratio. And why is this important? Because as we just saw, if SRT modulates it because of the BOD coming in, it means that the biofilm itself is impacted by what function it's being asked to do. And if there's a lot of carbon coming in, that might have implications on the heterotroph prevalence, as well as the thickness of the film itself. So how does it impact operability? So what I'm showing here is a comparison of the same MABR operated with primary effluent on the left and trickling filter effluent on the right. Thanks to the plant configuration and staff, we were able to do this, where we ran it with PE for a while and then the trickling filter effluent. So when you compare the water quality of these two, ammonias are comparable, but the COD of the trickling filter effluent is extremely low compared to the primary effluent, exactly what you'd expect. So quite a contrast in the soluble COD to ammonia N ratio, going from 7 to 2.5. What did we see? So we had sort of hypothesized that if we think about this film conceptually, with a very high C/N ratio, we'll get a lot of heterotrophs on the outside doing the work of BOD removal, and very little in the way of AOB, NOB, or nitrifier populations. In contrast, with a very low amount of carbon, we should see quite a low fraction of heterotrophs in the biofilm itself. So we tested this by operating with both influents, the primary effluent as well as the trickling filter effluent, at the same suspended SRT. And here, so there's a lot going on in this plot. Not just are we looking at what's being treated, and those are the shapes. The circles are treatment of primary effluent, and the triangles are treatment of the trickling filter effluent. So circles, high C/N ratio, triangles, very low C/N ratio. But layered on top of that, because of the way that we operated, particularly for the trickling filter operation, right, we had the opportunity to test two types of biofilms. The ones that have the yellow outline and the green inside triangles, those were the biofilms grown on, if you will, the primary effluent, and then switched over to treat the trickling filter effluent. And the gray triangles are when we started up a new system, a new biofilm, so a new membrane on the trickling filter effluent itself. Both, run for long enough that we can make this comparison between the data. And this was quite interesting for us. So the green triangles align pretty well with the red circles, very high ammonia removal rates, in the 3 to 4 gram N per square meter per day. In contrast, the gray triangles are much lower. So what this really told us is that, yes, the history of how the film is developed is very important. And we saw this visually as well. And it's so important that it could have the rate at which ammonia removal happens across the MABR itself. Keep going. I see there are some questions. I'm going to come back to them in a moment. The other thing that we did have an opportunity to test is what happens when you overload the membrane with organic carbon. So we ran with very high soluble COD loading rates for a while, and these are shown on the left. And then we ran same same setup with a lower soluble COD loading rate on the right hand side. And you can see, if you look at the left, early November, the film starts, it's okay. It looks dark brown and like a rich biofilm. And then by the time you get, 3 weeks or so later, it is sticky, congealed, gelatinous, lots of EPS being secreted. So very high loading rates, not a very good thing for the COD loading rates, excuse me. Not very good use of the membrane aerated biofilm reactor technology. And yeah, so this is essentially showing the same thing with the soluble COD loading rate increasing from bottom to top. And you can see that as you go to the highest panel, the one on top, there's practically no ammonia removal across the MABR zone, but with a comparable system but much lower soluble COD loading rates, you can get quite good ammonia removal across the MABR itself. So, I talked earlier about the unique characteristics of the counter-diffusional film, and this may induce some very interesting behavior. And the way we manage this is by a scour, so through a regular air scour. And this is an operational issue, as well as then resulting in some costs of doing that too. And so we learned, the impact of it is quite significant. What you're seeing here is in the effect of the rate of scouring. So from 80 to about 120 cubic feet per hour. That's the scouring rate, and how that impacts oxygen transfer rates. And then the two data sets are the frequency. So the things that you control are the intensity and the rate, or the frequency, and then how long. How long do you scour for every time you do scour? And that we didn't change, but we did experiments where we scoured every 2 hours versus every 4 hours, not looking at the performance, rather looking at the use of oxygen, and how that is influenced, right? So you can see that if you scour less frequently, you will drop the rate of oxygen transfer across the MABR film itself. So a less productive breathing film. So our key takeaways, related to the three questions that we were asking as a group. One, yes, indeed, we can do total nitrogen removal. We just have to, manage the suspended SRT and manage the balance between what the biofilm is doing and what the suspended growth system is doing. And some of that comes into how you would design the system, but also how you would operate it, perhaps seasonally. And I think one of the most interesting things we observed and learned is about how the the initial operation, so where you grow what you use to grow the biofilm, how that influences the performance and productivity of the MABR technology, and as a result, the value. We tried to sort of encapsulate what we learned into a simple logigram for us. And so the three key things that we see are, okay, where do you put it in the process? What is the suspended SRT that you pick, and how do you design and operate the scour to achieve effective biofilm performance and operation? And all of these, of course, end up impacting, in the case of nutrient removal, the nitrification rate itself, which is really the measure for us in terms of how well it's doing. I want to now just think about how we used some of these data from the pilot. So we did a few case studies. I want to briefly share one of those with you, and then jump into Q&A. I'm excited about that. So consider there's an existing treatment facility. It's quite old now, and looking to add assets and or be modified for the next 20 years. There are two effluent end-use opportunities. One is environmental discharge into the bay, and then the other is for non-potable reuse. It's a fairly large plant, it's about 20 mgd-ish, and has a pretty conventional characteristics in terms of the water quality itself. The kind of base concept is to build an entirely new facility of some size to accommodate it, and then get rid of the old one. So we are asked the question: if we were doing this as an MABR suspended growth system, how would this influence the size or operation or other aspects of the design itself? So here's the conventional approach. It's an MLE process. That was the base design, targeting about 10 milligram nitrogen per liter in the effluent. So I'm going to the conventional design was about 11 million gallons of total volume. In contrast, we found that with an MABR suspended growth design, using the model that we calibrated and developed and calibrated with our study data, it would be just shy of about 9 million gallons. So you could save 2 million gallons, and in the Bay Area, that's quite a lot of money to have an opportunity to save. So great. There's no question. There's some SRT and volume reduction, but we then asked, okay, what else would one have to be thinking about, right? So as I said, you're going from about 11 million gallons. This is the conventional MLE design, down to around 8 or 9, in that range. But the interesting thing is, you're going from a pretty conventional, 35% anoxic tank to a 50% anoxic to fit the MABRs into the design itself. So that's a little bit of a change. One has to just get their heads around that, and it makes sense. Where you're saying, look, the biofilm is doing so much work that I can make the fraction that's the suspended growth aerobic smaller than I would normally anticipate. Again, 2 or 3 million gallon savings in concrete is fairly substantial. Here are some of the ancillary benefits. Of course, said we're all striving for more sustainable nutrient recovery. MABRs offer better oxygen transfer efficiency than our conventional systems. So, not unexpectedly, we see a reduction in the use of air. So the blower, battery, the piping systems, everything can be smaller. That's a cost saving. The interesting thing to be thinking about is that there's also a reduction in the methanol savings. So supplemental carbon in the conventional approach would have been used to support denitrification. But with the MABR suspended growth strategy, you wouldn't need any methanol at all. So not only is that a cost savings, but methanol is not the most suitable chemical to be used at a plant. No, we've been using it for many years, but we're transitioning, of course, to other organic carbon sources. So in this way, you can avoid an entire chem feed facility or flammable chemical or any other supplemental carbon needs. So indeed, there are savings with concrete, and one of the things to be thinking about is you can actually, by reducing the volume, maybe keep the footprint the same, but reduce the side water depth of the suspended growth system. So it gives you a little, instead of having 25, 26-foot deep tanks where if you're doing maintenance staff are down there, you can have something shallower. So of course, the extension of all this is, well, can I just design and plan a conventional facility now and just plan to drop in MABRs? This is what Sandeep, you said at the outset that intensification is about using that infrastructure. It is. The only thing we have learned is that in order to get best value from the technology, the wraparound of the process has to be thought about beforehand. So if there's an idea that this can be expanded or intensified in the future to incorporate an MABR, some of that needs to be studied early on so that the hydrodynamics and the hydraulics of the system itself are conducive for plug and play in the future. Our data from the pilot and our collaboration with Hayward and Suez really, I think, show the value proposition of the technology itself. We are at this point quite content that we've answered some of our questions. Of course, like all, I'll say, good research. Maybe we now have maybe better questions than we had based on our work together, and we're actually very graciously Hayward has allowed us to stay on, and we continue to do research with them in other intensification strategies to understand how do all of these stack up. That is the end of my presentation. I'm very happy to answer the questions in the chat. And any more that there are or come along. I think you can use the chat, and I can get to it, and I'm going to start that now.
Excellent. Thank you so much, Sandeep. Yeah, if you want to ask a question, you can just go to the left-hand side of your screen where it says attendee chat, and you can just type it in there. And we already see two, so you can go ahead.
Yeah, that's great. What's the rounds number cutoff? Good question. I don't know that we specifically calculated what it was in the pilot itself, but like any other wastewater treatment process, they run in largely turbulent regimes. I'm trying to think about the context of the question. If it were very laminar, you'd get very little in the way of shear on the biofilm, and that's probably doing something to help renewal and just self scour, if you will, of the film. So you probably don't want to be in the laminar range. Do we consider Navier-Stokes? I'm not sure that I fully understand the second question. If you don't mind, can you expand on it a little bit more, just to say what it is that you're trying to fit? That's the part that I'm not sure I've understood. What index tells one when the ammonia is used up and ready to be changed? So, I mean, you can measure. So, we in the pilot or even in a full scale system, you would have ammonia probes across the reactor itself. And so, I want to go back. Excuse me. I'll go back a few. I'll just go. I'll go to the beginning and go forward a little bit. So, if I look at the pilot system, sorry, I'm going to just go there. Bear with me a moment. Next one. Here we are. So this is a flow schematic of the pilot on the left and the photograph on the right. We had an ammonia probe in the what's shown as the brake tank, essentially measuring the ammonia concentration in the feed. We also had one in zone 3, which is the aerobic zone, so just downstream of the MABR zone itself. And then in situ, we sampled each of these reactors. So you'll know, what the ammonia is doing across it based on the online measurement. So you're not really changing the membranes themselves. They're there for 10, 12, 15 years, whatever the life of the membrane is. So you don't really change the membrane itself. Great questions. Are there others?
Okay, I don't see any other questions.
Right.
Like I said, if anybody has additional questions, feel free to email Sandeep directly, or you can email me
Yes,
and I'll
please
be in
do.
touch. Great.
This will be, I think, uploaded, right? There's one question from
Yes,
Alex.
it'll be uploaded. The recording will be uploaded as well as the PDF. And I do see one more question.
Yes, I see that. Great question. We operated at a relatively short HRT of about 5 hours total HRT. So it was quite short HRT.
Okay, excellent. So we'll wrap up for today, and our next webinar will take place on Wednesday, May 18th at noon Eastern. This webinar is titled Reinventing the Toilet for Global Sanitation, the New Generator Resource Recovery Machine. It will be presented by Dr. Daniel Yeh, who is a professor of civil and environmental engineering at the University of South Florida. You can go to aaees.org and go to our events page to register. And just a reminder, if you're not yet an AAEES member and you're considering joining the Academy, please email me to discuss our membership options. My email is mwaterman@aaees.org. And like I said, we'll have a recording of this event up on our website by tomorrow, and I will be sending out PDH certificates this week. That's all for today. Thank you so much, and have a great day, everyone.
Thank you.